My name is Gangning Liang. I'm from USC, and today I majorly talk about the DNA methylation alteration in the bladder cancer. The bladder cancer is the fifth common cancer in the man and the sixth for the woman, and mainly found in the smokers. And bladder cancer also is one of the high recurrence type of cancer, and which required frequent and invasive monitoring. And just like all other types of cancer, both genetic and epigenetic alteration also involved in the bladder cancer. Like a genetic alteration, like a DNA mutation, the previous several people present, there also the epigenetic alteration, such as DNA methylation. That's what I want to present today. So in the normal cell, and usually the CpG side be methylated, but in the human genome have two types of distribution of the CpG. One is a group of the CpG we call CpG rich, or we call CpG island. Another is a CpG poor regions. So in the normal and usually CpG poor is methylated. And if this kind of methylation CpG site located promoter region, they may can silence the gene expression. And in the cancer, this kind of DNA methylation pattern is changed. Now they switch to the CpG island, and some of the CpG region will lose the methylation. The consequence is this one will also alter the gene expression pattern, and the consequence for the tumor will be maybe silence some tumor suppressed gene and react some of the oncogenes. So in this study, we do the large scale working on over 150 tissue sample from the blood cancer patients, and we use the methods is called Illumina Golden Gate DNA methylation array. This is a high throughput assay, and they can check over 700 gene the methylation status. And this 700 gene, the company picked the most gene is related played a role during the tumor genesis. So in this one is the heat map. You can see the yellow is represent methylate, and the blue represent unmethylate. On the top, you can see is I label different group of the sample. On the top of the left, NN, that means we call the really normal sample. That's the tissue we got from the patient without the bladder cancer. And the next is adjacent normal. That's the we also call the normal appearing normal tissue is from the patient with the bladder cancer. Next, we based on the stage, and we have a non-invasive tumor is from Ta, T1, and invasive tumor with T2 to T4. The last row is we put the ES cell, because we also want to see this kind of methylation change is that somehow mimic the ES methylation pattern or not. So in this study, we found this data clearly show two groups of the hypomethylation across all the tumor, regardless if they're invasive or non-invasive. The top one, this methylation pattern seems mimic the ES cell methylation pattern here. And the bottom one, it looks like really like a de novo methylation. This region never methylated before. And the second part we also found the hypomethylation, means lost methylation. And this one shows us another unique pattern that most show up in the non-invasive tumor. It's a low grade. So as I mentioned to you before, this blood cancer is a high recurrent. So people always think the normal appearing or the adjacent normal tissue maybe already show the pre-malignant change, or people also call maybe potential field defect. What we do here is we compare 34 match set adjacent normal with the tumor. We also compare with 11 the normal tissue from the patient without blood cancer. You clearly see the tumor is hypermethylate, and surprisingly, you also see the adjacent normal. A lot of region already gained the methylation. So this indicates this hypomethylation maybe played a role for the recurrence of the tumors. So the conclusion is, and blood cancer usually, we think a potential model is the first normal appearing cell, maybe pre-malignant change, and the next they go two pathway. 80% of a blood cancer is non-invasive one, and 20% is invasive tumor. And genetic alteration has been found in these two pathways. So they already found FGFR3 is mainly mutated in the non-invasive tumor and the P53 on the invasive tumor. Of course, some of the low-grade tumor can gain the P53 and become the invasive tumor. And our studies show the hypomethylation actually occur in both types of the tumor. And furthermore, we found that we think is very exciting is the field defect, or the normal appearing already showed hypomethylation. We think that's maybe played a role for the recurrence of the tumors. The final thing is the hypomethylation. They distinguish these two non-invasive and invasive tumors. So because we're very excited this result, we think this result have a clinical useful and benefits for the patient, because we can detect this methylation change in patient's urine. So right now, most assay we monitor patient recurrence is invasive assay. So we can use non-invasive assay and monitor for the tumor recurrence in the patient. We also maybe can screen for the bladder cancer in the high-risk population, like the smokers. So that's the data I want to share with you today. Thank you.